So now that we have established that intermediate filaments play a very important role in cell structure, let me tell you a few things about what happens when these cytoskeletal elements are not functioning properly. More specifically, mutations in the genes that code for intermediate filaments. Let's talk about that. Defective keratins expressed in epidermis cause epidermolysis bullosa simplex, in which skin blisters in response to even slight mechanical stress, which ruptures the basal cells. Other blistering diseases, disorders of mouth, esophageal lining, and cornea of the eye are caused by mutations in different keratin genes, which are expressed specifically in these tissues. Typically, cells rupture as a consequence of mechanical trauma and a disorganized clumping of keratin filament cytoskeleton occurs. Many of the specific mutations that cause these diseases alter the ends of the central many of the specific mutations that cause these diseases alter the ends of the central rod domain, underlining the importance of this particular part of protein for correct filament assembly. We have the micrographs. So this micrograph is of a normal, it was taken from, is a cross section from the normal tissue, the one I'm going to highlight here, this one. You can see the tissue in response to slight mechanical trauma has maintained its integrity. The one on this side is taken from an animal which was expressing truncated keratin protein lacking both the N and C terminal domains. This was expressed in a transgenic mouse. The defective protein assembles with the normal keratins, thereby disrupting the keratin filamental network in the basal cells of the skin. So basically, this mutant version is acting again as a dominant negative. Here you can see the mouse which is expressing the truncated version of keratin filaments. There is a blister forming in response to a slight mechanical trauma. This is what happens. This is a diagram of what happens. So basically, when stress is applied, force is applied in this direction, the intermediate filaments whose job is to provide structural strength, they break. When they break, the cells come off, as you can see here, and that loss of cell basically results in formation of different blisters. So these are the defective keratin filaments. And keratin, as we have said, is a type of intermediate filament. Three types of neuronal filaments that we have talked about, NFL, M, and H, they co-assemble in vivo, forming heteropolymers that contain NFL plus one of the other. These neurofilaments are found in high concentration along the axons of vertebrate neurons. The NFH and M proteins have lengthy C-terminal tail domains that bind to neighboring filaments. This generates uniform spacing between these filaments. The neurodegenerative diseases, amyotrophic lateral sclerosis, abbreviated ALS, or Lou Gehrig's disease, is associated with an accumulation of abnormal assembly of neurofilaments in motor neurons and their cell bodies and also in their axons, which interferes with the normal axonal transport, thereby inhibiting the function of these neurons, motor neurons, which basically, when are activated, result in contraction of the muscle. So this happens in these diseases, and the root cause is basically that their intermediate filaments are defective. Here you can see these are the freeze-etch electron micrographs. In the neurofilaments, in neurons, they have these proteins which are associating with these filaments, and they form these extensive cross-linking of these filaments. Not only they're cross-linking, increasing the structural strength, they are also spacing these filaments at a specific distance. Here in this micrograph, electron micrograph, you can see that these filaments, intermediate filaments, do not have these extensive cross-linking because the type of cell that is producing them does not need that kind of structural strength. And these are taken from glial cells, and these filaments in glial cells are not cross-linked. In the next micrograph, I'll show you, this is basically conventional electron micrograph, a cross-section of an axon showing the regular spacing of neurofilaments. So these dots, tiny black dots, are neurofilaments. You would appreciate that how they are evenly spaced in this axon. And also these hollow ring-like structures are basically cross-sections of microtubules. So again, they're hollow, as we know. So these neurofilaments are spaced appropriately in the tissue, and which is the function of the proteins that are associated with the neurofilaments. They not only space them, they also provide structural strength, since they are linking intermediate filaments or the neurofilaments in the neuronal cells.